All right, we're recording, and now we're going to go into our lesson. And today we're going to talk about physiological foundations for psychology. And the same way we talked about philosophers that shaped or influenced the development of psychology, now we're going to move on to physiology. And the concept of physiology and its relationship to psychology is a long one as well. So there's been speculation from even Greek philosophy as to where the mind operated and worked. So if you look at people like Aristotle, 2,300 years ago, he believed that the heart was the seat of the mind. And you might see references to this, follow your heart, things like that. And that's bad advice, because we know today the heart is not where thinking occurs. It's not even where emotion occurs. All the heart does is pump blood through the body. But nevertheless, there was this assumption. Now, Galen talked about the four humors, and he felt that the four humors operated through the through the ventricles. And again, wrong, right? Even the sense of the four humors, which were black bile and yellow bile and blood and phlegm, these four fluids, he thought were related to somehow the ventricles. And if there was a dysregulation of the four humors, then that would result in some form of psychopathology. Now, he also talked about the concept of spirits flowing through the ventricles. And we see that certainly influenced Rene Descartes. And remember how we talked about in the last discussion about Descartes and how he felt animal spirits flowed through hollow tubes, right? Now, we know that's wrong as well, but nevertheless, he was influenced by Galen. Ultimately, we start to see a shift towards the brain as being the focal point in the 1600s. So Willis came to the conclusion that the brain was the center of the mind. And if you were to look at this, you start to see how this shift works. So Aristotle with the heart, then Galen with the ventricles, the lateral ventricles, third and fourth ventricles. Then Descartes moved to the pineal gland, and then by the time we get to Willis, we see the brain as a general functioning. Now, we understood the brain in, I want to say, a gross anatomical way, or brain regions to some degree, but we didn't really have an appreciation for the more micro level. So the macro of brain structures we understood, but with Golgi was, he came up with a staining mechanism that you could actually isolate a single neuron and visibly see the dendrites, the soma, and axon, which is pretty cool, right? And then by the early 1900s, we were off to the races trying to measure action potentials and responding of a single neuron. And now we understand the neural network, that multiple neurons come together, they fire together, and the brain sorts out all of the electrical impulses. So, but if we're talking about physiology, we can also talk about the concept of measurement error, right? Because if we're relying on someone's perception, it is possible that two people might perceive the same experience in slightly different ways. And we saw this. We saw this with Reverend Neville Mescaline. He was a British astronomer, and he studied the speed by which a star moved from one point in the sky to another. And he had an assistant, Kinnebrook, or David Kinnebrook, and David Kinnebrook took measurements of the amount of time it took for a star to move from one point in the sky to another. And what he found was that Kinnebrook's measurements were slower than Mescaline's by anywhere between a half a second to almost a second. And Mescaline got upset, and he felt that Kinnebrook wasn't really taking this seriously or accurately. So he fired him. Now, the lesson here is that there are individual differences, right? So two people are going to experience the same thing, but our reaction to a sensation, our reaction to processing a perceptual experience is going to be different. So we have to acknowledge individual differences between observers. Now, just to give you a sense of the Royal Observatory, here on the left is a sketch of the Royal Observatory, and on the right is an actual photograph of the Royal Observatory. And this was, a very, very important aspect of science in the British world. And there are observatories throughout the world. I don't know if you know, but the College of Staten Island actually has an observatory, and you could actually track stars as they move through the sky and see how your times are relative to Mescaline and Kinnebrook. But I doubt you're going to do that, but I figured it would be cool to say. So 20 years later, Frederick Bessel comes onto the scene, and he tries to understand how was it possible for Kinnebrook and Mescaline to have such disparate measurements. And he started to explain the concept of reaction time. And reaction time is the amount of time from the presentation of a stimulus to getting a response. And remember those individual differences that we talked about. Those individual differences, well, guess what? The explanation of these individual differences are due to differential reaction times. And he was able to calculate, he created what's called a personal equation, where he could calculate these individual differences and do a correction formula that would eliminate some of the error in the data. So ultimately, if you are taking research methods, you might hear that every observed score is a combination of a true score plus error. And these individual differences in reaction time play a role in error and the differences in these scores. Now, what did Bessel conclude? He concluded that we have to consider human nature. We have to consider individual differences, because we all operate slightly different based on our perceptual abilities and personal traits. And there's always going to be some element of error. And we have to account for that and acknowledge that. Now, this isn't just astronomy that has this problem of error. It's every science has some error, especially if you're using observational research strategies, because there is a subjective element from person to person. And I think that's important. Now, science does not expect 100% precision. If you think about it, Mescaline was a little bit harsh on Kinnebrook, because if we were doing a statistical analysis, we set our alpha in general at 0.05, which means that we're allowing in this study up to about 5% error. And that's all sciences: astronomy, biology, chemistry, physics, psychology. It doesn't matter. The standard alpha is 0.05. So we acknowledge there's going to be some error. And that's how we address it today is, hey, as long as the amount of error in this study is minuscule, we're okay with it. And if it's less than 5%, we're okay with that. Now, does that mean every science operates based on an alpha of 0.05? No. If you do pharmaceutical research, you're going to be far more stringent in how much error you're willing to allow. And the reasoning is pretty simple. If you're giving people medication, you want to take as little chance of harming them as possible. So generally speaking, medication-based studies, we set the alpha at 0.001, which is the error has to be less than 0.1%, or I'll say it differently, less than 1 out of 1,000 people are affected adversely by this medication. Now, because most drug trials are with about 1,000 people, less than 1 out of 1,000 means that no one in the study was harmed, practically speaking. So, but your science, depending on what you're studying, you might tighten up the alpha, be more stringent in your alpha. All right, so valuable lesson from Bessel. And I think if we go back to the philosophical antecedents to psychology, we already said this, right? So if we look at John Locke and George Berkeley, they acknowledged that perception was very subjective, which means that it's a person-to-person basis, and we can't assume that my experience and your experience is going to be a perfect match. We also can't assume that our perception of something and what's happening on out there in the real environment is going to be a perfect match. So we talked about this, and many philosophers, many researchers acknowledge individual differences in error. Okay, so now we're going to talk a little bit about physiology, right? We talked about the brain, we talked about the heart, and we're going to talk about the processes by which we perceive an experience. So sensation, perception, and that was the focus of the day. In the 1700s and 1800s, there was a massive boom in biological research, or neuroscience research, as we might call it today. But this research sets the stage for psychology. So between the theoretical that we talked about from philosophy, moving to the empirical observations, and then the physiology combined together to bring us to the end of what we'll mention at the end of lecture 3 about Wilhelm Wundt. Wilhelm Wundt did not happen in a vacuum. The philosophy, the empiricism, and the physiologists all came together to make that moment. Rosa Maria, you have your hand up.
Yeah, I'm sorry, Professor. My computer wasn't working, but I'm here. I don't want you to think I didn't show up.
No, I take attendance twice, so you'll get partial credit. Don't worry.
Okay, thank you.
Sure. Okay. So now, when we talk about physiology, physiology is basically looking at a stimulus and a response in the brain. So, and measuring the effect of that. So if I present a light in the visual system, what happens in the brain, right? Now, psychophysics is more about the mental interpretation or the perceptual experience. So if I present a light and I ask you, what are you seeing? And you say, I see a red light. Your perception of redness is more of a psychophysics question, whereas physiology might be neurons in the visual cortex firing, so to speak. So let's start off the Belmonte Law, right? So Charles Bell and Francois Mejonde, they realized that in the spinal cord, there are two nerve endings. And they actually were able to discover that there's a dorsal root and a ventral root, and sensory nerves enter through the dorsal root, motor neurons fire and go out or enter through the ventral root. So if you look at a cartoonesque depiction of a spinal cord, you're going to see like projections on two parts. Now, the dorsal root also has a bundle of fibers called the dorsal root ganglion, right? That's all a ganglion is, a bundle of fibers. But then that fiber sends a fiber track all the way up to the somatosensory cortex. But Charles Bell discovered this, and Mejonde discovered this. They didn't work necessarily together, but because they're somewhat simultaneous discoveries, we tend to refer to this understanding as the Belmonte Law. So the Belmonte Law suggests that there are two types of nerves: sensory neurons, motor neurons, or sensory nerves, motor nerves. And sensory nerves carry input from a receptor site all the way up to the brain, and motor neurons carry impulse from the brain down the spinal cord and out to the muscles and the glands in the body. That was a remarkable discovery. They got it right. And in fact, if there is a person who's experiencing pain, one of the treatments is to sever the dorsal root ganglion so that the pain impulse won't go all the way up to the brain. So there are different applications clinically of what Bell and Mejonde found, but it's pretty impressive that they found it in the late 1700s, early 1800s. So Johannes Mueller, he's another major player when it comes to the study of physiology. And he really took a scientific or experimental approach to try and understand physiology. Now, he was a very prominent figure, held a position at the University of Berlin. In Germany, the University of Berlin was kind of like their Harvard. So it was an impressive position for him to obtain. And he was, an eminent scholar. And he published an article every 7 weeks for 38 years. Now, ultimately, putting that much pressure on yourself is intense, and it can induce anxiety. It can induce, negative self-talk, and he develops depression and eventually ends his own life through suicide. Now, when we look at his writings, he also wrote a handbook. At this time, writing, not only did they write journal articles, but they wrote books. And in his handbook, he talked about his theory of specific nerve energies. And the premise of this was that different neurons induce different sensory experiences. So based on that, if we were to think of our five senses, each sensory nerve has its own specific nerve energy, and that activates the sense organ or sensory neuron. And that makes sense, right? So he got that right for sure, right? So if we were to think of vision, well, what is it that activates vision? Light waves or electromagnetic waves on that color spectrum. So that's going to hit the back of the retina or the more peripheral rods, and it's going to induce some kind of visual experience. In our hearing, there are sound waves that create vibrations in the cochlea, and there's this shearing effect that creates a sensory experience that sends up to the brain. And touch, we have skin receptors in our skin that are able to detect painful stimuli, non-painful stimuli, and so forth. So he definitely got it right. Now, he used the term called adequate stimulation. That term was meant to say that is the maximally sensitive stimulation for that sense organ. So we talked about light waves and sound waves and things like that. But we can also induce a sensory experience with other forms of stimulation. So he acknowledged that there are less adequate stimuli that could induce a process. So if you were to close your eye and press on your eyeball, you'll probably see spots, right? And what is the flickering white lights that you see? Well, you're actually stimulating neurons, even though your eyes are closed. So that physical pressure that you're putting on could activate a visual experience. But that's not, the typical. So we tend to focus in sensation and perception research on the adequate stimulation process because, we want to understand how it naturally occurs. So what were his contributions? Well, number one, he was able to understand localization in the brain. That is to say that different parts of the brain have separate and distinct functions. That's a huge thing to understand, that the different parts of the brain do different things. He was also able to identify your sensory receptor mechanisms. Now, he didn't get it all. There are researchers who fill in the gap after him, but to understand how sensory receptors work and to understand localization in the brain, that's going to propel further research that fills in the gaps, as I say. So now we start to try and map the brain, right? So Marshall Hall, which was a Scottish physician, starts to study how the brain works with decapitated animals. So he would start to stimulate decapitated animals and see what kind of physiological response he would get. So depending on where he stimulated, he was able to determine certain features. So voluntary movement was found in the cerebrum. Reflex movement was found in the spinal cord. Today, we know about the reflex arc that is there. Involuntary movement could occur with direct stimulation of the muscles. If you've ever done a neurological test, you know that is the case. And respiratory movement occurs in the medulla. Now, the medulla, it's interesting when you think about the time period. I'm, in addition to being a clinical psychologist, my master's is in neuroscience. So when I think about what all these scholars were able to do, I'm impressed because they didn't have the sophisticated research tools that we have today, but they were able to identify things. We know that the medulla is responsible in all vital functioning, breathing, heartbeat, digestion. So for him to understand in a dead animal what the medulla does is pretty impressive. Okay, now there are other ways to study. So there was the decapitation method, which we talked about. There's the extirpation method. Now, the extirpation method is basically a way of either destroying or by lesioning or removing a part of the brain or body and seeing what happens. So Pierre Florenz used pretty commonly the extirpation method. Now, Marshall Hall that we talked about also used this method. And what did Florenz find? Higher mental processes, cognition, things like that operate in the cerebrum. Now, that's true. It's vague, but true. Visual and auditory reflexes operate in the midbrain. That's true. We've identified both the inferior and superior colliculi as visual and auditory reflex functioning. And then heartbeat, respiration, vital functions in the medulla. So he elaborated on what Marshall Hall did. And he wrote what he could. And everything he wrote was actually correct. Motor coordination in the cerebellum, quite impressive when you think about it. Now, so he was able to argue that there was localization in the brain. Ultimately, Florenz did believe in integration centers or integration areas, and he basically felt that the brain operated as a unit, a whole unit. That he didn't get entirely correct, because there are localized brain regions that do specific things, and there are integration areas in the brain. So he was partially correct about this concept of integration areas, but because it was exclusive, he thought it was purely an integrative unit, he didn't get it entirely correct. Now, Franz Gall, you'll hear about him, but his claim to fame is the concept of hrenology. And he went to the opposite extreme of the brain having specific areas, and each area has its own functioning. Now, I'm gonna go get my phrenology head, and I'll be right back. Hang on for a second. This is my showpiece. I don't know if you can see, but he labeled all of the different areas and what they did. And I'm going to slowly turn it around so you could see that he believed that there were certain functions of different parts of the brain. Now, all this being said, yeah, there is localization in the brain, 100%. But every label virtually that I showed you as I rotated the this head was wrong. He got most of it wrong. So you might say, well, why are we going to be talking about Franz Gall? If he got it all wrong, does it make sense that we should waste our time? The answer is, of course we should talk about him, because even if a person gets something wrong, it inspires other researchers to test that understanding, and ultimately inspired research on localization of the brain. And we have a really good understanding of the concept of localization in the brain today. So here we go. Now let's talk about Paul Broca. Most of you have heard the name Paul Broca based on the concept of Brocas Area. So Brocas Area we'll talk about, but how did Broca understand or do research on the brain. So he used what's called the clinical method. And he basically took patients' brains after death, and he examined where there were any damages. And what he did was he correlated the damage in the brain with physical illnesses. Now, this connection is not a perfect system, right? Obviously, when you have a person with a brain injury and you correlate it, you could miss things. But this, in addition to the extirpation method, is quite helpful because we can study both while you're alive as well as when you're, when you're dead. We're able to study. So there were all of these different research strategies out there to try and understand what's going on. Now, you probably have heard of Brocas Area. I don't think you can be a psychology student without hearing this name. If you took it in the introduction to psychology, you would have heard about it. If you took developmental psychology, when you heard about language development, you would have heard his name. If you took neuroscience, you would have learned his name. It doesn't matter. It's a pretty common name in the books. So what did Broca find? Paul Broca discovered that there was a specific brain region responsible for speech and language production. So if you were wondering how it is that I am capable of lecturing almost reflexively without even thinking, right? It's because I have a functional Brocas area. So that allows me to articulate, that allows me to speak. Now, what's cool is that even though the Brocas area is found in the inferior frontal gyrus, it's right under the motor cortex. And that's important because the motor cortex is what's responsible for sending a message from the brain to the periphery. So while Brocas area is responsible for speech and language production, I need to be able to move my mouth. I need to put proper placement of my tongue. And for me to do that, there needs to be communication between Brocas area and the motor cortex. So altogether, that's what it is. Now, Paul Broca found that Brocas area is generally the opposite side of your dominant writing hand. So if you are right-handed, Brocas area is on your left side. Whereas if you're left-handed, it's interesting, only about 50% is it still on the left side. The other 50%, we have something called codominance. And I say we because I'm also left-handed, but we have codominance. So we're going to use part of the inferior frontal gyrus on the left side and the right side. And you might say, well, big deal, so what? You're sharing dominance. Well, that's an important benefit for left-handed people if you have a stroke in that area, right? Because if you have a stroke in the inferior frontal gyrus, you're going to lose your ability to articulate and produce speech. Well, if you have codominance, then you're going to be less likely to lose it because the right side will kick in. So that's Brocas area. That's the concept of codominance. It also highlights what we talked about as localization in the brain. Now, based on this principle that the left inferior frontal gyrus is the center for speech and language production, that seems to push against Floren's idea of an integrative unit, right? So I give Floren's a little more credit than is on the slide because we do have association areas, right? Integration areas. But to suggest the brain works as a a unitary brain. Broca's research and many subsequent researchers have pushed back against it and disproven it to a great degree. So here's a cartoon picture of what a brain looks like, and highlighted in purple is roughly where Brocas area would be. And if there is damage to Brocas area, you would have an expressive aphasia. So remember how I said you would have the inability to articulate properly? That would be a form of an expressive aphasia. So Fritz and Hauser, this is our next major players. And Fritz and Hauser are absolutely critical when it comes to neurophysiology. So their methodology of electrical stimulation is going to be used, and you're going to see multiple subsequent researchers who use this strategy to map the brain. So what is electrical stimulation? You take an electrode, you put it at different parts of the brain, you pass a minuscule electrical current, forcing the neuron to fire, and then we measure the motor response. And Fritz and Hauser did not map out everything, but what they were able to suggest is that different parts of the motor cortex are responsible for different motor activities. So if I stimulate one part of the motor cortex, maybe my arm would raise. If I stimulate another part of the motor cortex, maybe my leg would twitch, and so forth. So that was the conclusion that Fritz and Hauser did. But along comes Ferrier and Penfield. These individuals were crucial for mapping out in a more systematic way what different parts of the somatosensory cortex did. So David Ferrier used electrical stimulation to determine what place in the skin is located in the cortex, right? So linking different parts of your skin to the somatosensory cortex, which is responsible for touch. So he added more to the mix in terms of mapping the somatosensory cortex. But it was Wilder Penfield who we give the most credit, because he systematically mapped both the somatosensory and the motor cortices to identify each part and how it fits in with the body. Well, technically the face and the body. And what he found was really incredible. If you look at, on the left, the somatosensory cortex, and if you look at, on the right, the motor cortex, you're kind of looking at almost like a person hanging upside down on the monkey bars, right? With their legs up and wrapped downward is the rest of their body. And that's what he found, was that there was a systematic representation of the face and body in a continuous way. So just like we have our from head to toe, in our actual body, in the brain, it's very similar. And this is what he referred to as a topographic map. So he's able to map everything. You see on both sides, it's virtually identical. The one difference that is glaring is that in the somatosensory cortex, you'll see, and I'm going to move my cursor, that the feet and the genitals are very close together. And it's not easily explained as to why that happened in a sensory point of view, because it should be further up on the cortex. But it does explain how some people find foot massage erotic, right? So because neurons that wire together fire together. So you might, while stimulating the feet, also stimulate some excitement in the genitals. But neither here nor there, there's 90+% match in the motor cortex and the somatosensory cortex. Pretty cool. Now, the other thing you'll probably notice about this picture is that it's a distorted picture of a person, isn't it? Look at how much space, and I'm going to move my cursor again, is provided to the face on both sides, right? So he drew this in terms of the representation on the brain. So you might hear the term homunculus. So if you Google homunculus, it will show you almost like a 3D picture of a person, what they would look like if their physical shape matched the topographical map on the brain. It's not the case, but if you ask, well, why is it that your face gets more attention, or why is it that your hands get more attention and then things like your back get less attention? It's pretty simple. The more a part of the body is essential for survival, the more attention it gets on the brain. And our brain may look like this based on Wild or Penfield, but if we look at different species, their homunculus, their picture would look very different. So if you were to compare a human homunculus to, a dog homunculus, you're going to see far more attention to hearing for a dog than is for human beings, because hearing is more essential for vital functioning and survival in a dog model than a human model. But every picture you see of the somatosensory cortex and the motor cortex with these cartoons comes from Wild or Penfield and his ability to map out the somatosensory cortex. Now, today, the cortex has been entirely mapped out. So you could Google, and I didn't put it in the slideshow, but look at Brodmann's area. So Brodmann's area is the way that we mapped out the whole cortex. Penfield was most interested in the somatosensory and motor cortices. Okay, so like these are individuals who started to map out different parts of the brain and what they did. So you're seeing localization in the brain. But this also inspired a group called faculty psychologists, right? So faculty psychologists understood the concept of individual differences. If we go back to Bessel and how there could be differences in reaction time and vision, well, the same thing could occur in mental abilities too. So faculty psychologists made the assumption and stated that mental abilities do not exist to the same degree in everyone. Some people have better analytical skills than others. Some people have better spatial skills than others, and so forth. But what they felt was that in addition to there being this localization in the brain, we could actually quantify how competent this person was based on the bumps on their head. So if they had well-developed areas, bumps or protrusions, then we would say they have greater mental faculties in that area. Now, keep in mind, Franz Gall is one of these faculty psychologists. So if you're looking at the bumps on the outside of the head and mapping it onto some kind of functioning that's wrong, you're going to draw the wrong conclusion. But what he also suggested is that underdeveloped areas, areas that are not as large or did not protrude as much, were considered areas of deficit, where a person had poorer mental faculties. Now let's see. That is indeed a myth. It was entirely wrong, Jocelyn. So Jocelyn asked, wasn't that a myth? You are correct, but we are teaching the history of psychology, so we have to talk about the phrenologists. We have to talk about the people that are wrong. Now, today, since you asked the question, we know the bumps on your head don't even match the shape of your skull, let alone the folds in the brain. So this idea of mapping the outside of your head and trying to suggest it had some kind of mental ability, it was entirely wrong. And people paid big money to have their heads measured to see how competent they were. And you'll see a picture a little bit later. So you're 100% correct, Jocelyn. That was indeed a myth, but it permeated the field in the 18 and early 1900s. So let's talk a little bit about my man, Franz Gall. So he used the clinical method to confirm the concept of white matter and gray matter. That is true, right? So everybody knows when you see white matter in the brain, that's it's usually axons. Highly myelinated axons make the white appearance, whereas if you see gray matter, it's dense cell bodies. So he's able to see that. He was also able to see that nerve stimulation projects contralateral. So if a person, let's say, gets tapped on the left shoulder, then it's going to go to the right side of the brain. We know that with stroke victims, right? So if there is a damage to a part of one side of the brain, it usually affects the other side of the body. And he was also able to appreciate how the two hemispheres communicate with one another. Now, we talk about the corpus callosum, the anterior and posterior white commissures as the bridges between the hemispheres. So he confirmed a lot of these things, which is quite impressive. But when he drew the conclusion that larger brains resulted in larger intelligence, he felt that he should measure that. And in general, that is the case. There is a correlation between brain size and intellectual functioning. However, it's not a perfect correlation. So he took that conclusion and started doing cranioscopies, where he started to measure different parts of the outside of the head and draw conclusions about intellectual and emotional function. And of course, the localization of the brain, again, is relevant. But it became popular, and people, went through Europe and the United States giving lectures on phrenology, right? So Sperheim and Combe were the major advocates or the spokespeople for this concept of phrenology. Society started to develop, and people started using phrenology to select employees. And even when it came to couples counseling and testing children, people started to use it to draw conclusions. Now, Gall's research ultimately was discredited by Florenz, which we mentioned earlier. And we know that today the map of the head doesn't match the actual function. So whatever map they used or drew up, it was just entirely wrong. So Franz Gall went from being this really incredible scientist who made all those accomplishments that I talked to you about in the beginning to being viewed as, a charlatan, a quack, a fraudster, however you want to say it. But we still talk about it, even though, as Jocelyn asked, isn't it a myth? We still talk about it because we don't hide the warts in our discipline. We want to talk about it. But even when a person is wrong, if it inspires something that is demonstrated to be true, we want to know where that inspiration came from. So mapping the head, those phrenology head maps, inspired people to actually, after realizing he was wrong, determine what part of the brain did what in a more systematic way. So here is a picture. Again, another example. Now, if you see, I kind of brought my head to the camera, but you could see these maps. They're online, and the diagrams you could see, and different parts, the numbers and what they reflected. And it's almost comical today. But on the left, you also see what the cranioscopy looked like. So it was this it was almost like an upside down sieve, a metal contraption that you put over the head, and rods would point to different parts to measure how protruded it was. And that's how they drew the conclusion about your mental faculties. Mythology, falsehood, but it was very, very prominent. And can you imagine hiring people based on the size and shape of their head? Can you imagine deciding whether to admit a child into a private school based on the size or shape of their head? It's comical, but nevertheless, it was part of our history. I wouldn't get hired. I've got a small head. Yeah, but you know who would get hired? George Lopez. George Lopez could get any job he wanted. Those of you don't know, look him up. He's a really incredible comedian, but he makes fun of how big his head is. So I figured I'd make fun of it too. All right. So now let's start to discuss further electrical studies. Luigi Galvani, he was an Italian researcher who also studied the nervous system. And he came to the understanding that neurons had electrical impulses. And that was incredible, right? Because keep in mind, in Rene Descartes' time, he was talking about animal spirits flowing through these hollow tubes. Turned out not to be true, right? It turned out to be these electrical impulses. And if you remember Hartley talked about vibrations, almost like a string. I mentioned, like, if you pluck a string of a guitar, it vibrates. Ultimately, Galvani changed how we understand a neuron. And by the 1850s, we pretty much adopted this understanding that inside a neuron, it operates based on electrical impulses. He was right. And if you were to think about what you learned about action potentials, we talk about electrical impulses, don't we? And how the voltage changes based on hitting the threshold and the neuron fires. But we're measuring electrical current inside the neuron. But we've known this since the 1850s. Now, so Galvani and Aldini ultimately took advantage of criminals, and when they were dead, they used the severed heads to study muscle movement and demonstrated there was electrical current moving those muscles. So again, you're seeing all of these different methodologies that are used to try and get a better understanding of the body. Now, if anyone wants to go to medical school, you're going to be doing this. You're either going to have your own cadaver, or you might go to, what the hell is it called? It's a artificial simulation of a cadaver, a lab where you're going to be studying the different parts of the body and what they do and what kind of pathology occurs in these different parts and what it looks like. So you're going to be tinkering with the body as well. If you like what I'm talking about today, you know, you might even consider going to the bodies exhibit. I know some people might be grossed out by that, but it also is a pretty cool depiction of what our body looks like at different layers. Now, in terms of transmitting sensory and motor communication, we understand that the nervous system is responsible for that. So you might hear the term central and peripheral nervous system, but the nervous system ultimately serves as the switching station, the kind of controlling when a motor impulse goes out or when a sensory input comes in. Now, pretty cool. We pretty much have rejected Hartley and Descartes' understanding. And now, you know, we also understand that it's pretty much automatic. So the autonomic nervous system operates pretty much on a lot of this. So if you have a stimulus, let's say someone taps you on the shoulder, that is going to activate a sense organ. What is the sense organ? It's the skin. Random Jeopardy fact, the skin is the largest organ of the body, right? So if they tap you on the shoulder, you're going to have the skin, there are neurons there, and it's going to send an impulse from that skin wired all the way to the spinal cord, depending on where it would usually be it would probably be the thoracic level of the spinal cord. It would go across and then it would go up to the brain. So the impulse goes, hits the nervous system site, right? Goes all the way up to the somatosensory cortex. And then it says, okay, I've been tapped on the shoulder. I need to respond to this. Sends a message down to the neck. So you turn to the person, right? And it sends a motor response. So how it works. Now, very broad strokes, right? So we talked about brain regions, but now let's get on the microscopic level. A lot of what we know about what a neuron looks like comes from Santiago Ramon y Cajal. Cajal, his drawings are incredible, and I'm going to show you a couple, but ultimately he was able to understand how different sensory neurons operated and how the wiring looked like. So it's pretty cool. Eventually he wins the Nobel Prize, but I also want to talk about culture. You may remember in lecture one, I talked about how there were some biases against women, against African Americans, against Jewish individuals at this time. There was also a linguistic bias. If you looked at the journals were most often written in either English or German. And because Cajal, his writings were in Spanish, they were ignored for quite a lot of time. And that resulted in us missing out. When they're discovered, he does get recognized properly. But imagine we discovered this earlier. Imagine we were able to capitalize on his wisdom earlier. So what does this teach us? You know, there are certain groups that are disadvantaged in a given society because they are not part of the mainstream culture or the majority group. We see that with Lev Vygotsky when we talk about cognitive development. Everybody gives love to Piaget, but Vygotsky, you know, he developed an alternative understanding of cognitive development, but he wrote in Russian. And similar to Cajal, his work was ignored because it wasn't part of the mainstream culture, which is a shame. But here's a picture of Cajal's view of neurons in the cerebellum. You can see that he drew different kinds of neurons to demonstrate that each neuron has a different kind of shape. And you can see Purkinje cells and pyramidal cells depicted in these drawings. And that's important for us to understand, because if we compare those neurons and their shape, and I apologize, when I blew this up, it got a little blurry. You know, the drawing of the mapping of the retina, the rods and the cones connected to the bipolar cells and the ganglion cells. You know, he understood the relationship between these different parts of the neuron, of the visual system, which is pretty remarkable. And the neurons look different. And if you were to take a biological psychology or a neuroscience class, you will learn that there are different shapes of neurons, and the shape of the neuron impacts its functioning. So my screen popped out. Can you still see it?
Yes.
Okay, good. All right. So, but yeah, there are different neuron shapes, and remember, its shape and function are related. The shape and function are related. So you'll learn more about that if you take a biological psych class. And I do apologize that it's blurry. I did a what's it called? Collective Commons or Wiki Commons is a free image that doesn't have a copyright on it, but I didn't realize that when I magnified it, it got blurry. So I apologize about that. Now, in terms of the nervous system, today we have a pretty good understanding that the electrical impulse that we discussed so far is what happens within a neuron. But we started to become aware that these electrical impulses are not continuous. We once thought that the electrical wire ran from point A to point B. And then we discovered the concept of a synapse. So we realized that there were spaces between neurons. And then we had to figure out, well, how the hell is neurological functioning happening if we have these severed electrical wires? It should stop the process of communication. And then we realized neurotransmitters, and we started to appreciate the functioning of the chemicals in the brain and how they're released into the synapse and bind to another side. But really, when we think about the neuron and the synapse and all of these aspects, it's a very mechanistic point of view, right? So we can say, oh, here's the mapping of the structure of the brain, and we could even break it down to the brain's gears and gizmos. And if I use that analogy because in a philosophical lecture, we talked about the clock and all the gears. Well, what are the gears in the brain? Neurons and synaptic connections and nerve fibers. All of these things are the mechanical structure of the brain. We also, because we started to appreciate a neuron is just a fancy way of saying a cell. Cells are microscopic. They're very, very small, right? But you have roughly 100 billion neurons in your brain, 100 billion. So how do we get these brain regions? Well, we have a bunch of neurons that bind together and combine to become a structure. So the atomistic understanding of how cells bind together to become regions of the brain was also relevant. So think about what we said in terms of mechanism and the goal for reductionism or an atomistic understanding. Even though we're talking about physiology, it's still ever present. So this mechanistic spirit was pretty profound within Germany. And Mueller, which we talked about earlier, created the Berlin Physical Society, which he argued that the principles of physics are relevant and can explain virtually everything. And he wanted to connect the mechanistic physiology to physics. And today we've done a pretty good job. If you take anatomy and physiology, it does operate based on this mechanistic understanding. Think about how we have different systems in the body. Well, if you have different systems in the body, let's say the cardiovascular system, well, the cardiovascular system, we can break it down into its parts, the organs that are involved in a cardiovascular system. So he did a pretty good job pushing this. And then he only focused on what the blood oath was, to only focus on the active forces that we can study through physics or chemistry, physiochemical. Now think about that, right? When we talk about a neuron, it does operate based on a physicochemical process within the neuron versus between the neurons. So pretty good. So, and we get support, right? We get support from research. We have the British empiricists and even the French, right? They're pushing this sense of direct observation or direct experience. If we're talking about psychophysics, we would be talking about sensory experience. Then we had Bessel, which we talked about earlier, trying to understand how we have difference in numerical values. And he came to the conclusion that there are individual differences, and these individual differences occur because of sensory or perceptual experiences. And then we had the physiologists who basically mapped the structure and the function of our senses. So the next step was to experimentally measure subjective mentalistic experiences, was to measure sensation and perception. So when we talk about psychophysics, well, that really is at the heart of experimental psychology. And experimental psychology was what we did first. So when we talk about Wilhelm Wundt, he was an experimental psychologist. He studied consciousness, but he did it in an experimental way. And in lecture 4, we're going to talk all about what he did and the scientific rigor his experimentation took. But there are other major players like Hermann von Helmholtz. Helmholtz is, again, I hate to call him a Renaissance man because it's happening later than, say, the Renaissance, but he was a Renaissance man. He contributed so much to so many various areas. So we're going to talk about him. And then we're going to talk about Weber and Fechner, which you probably have learned all about them in your biology class, and then we'll get to Wundt. So you might ask yourself, well, why was Germany the origination of psychological science? Well, Germany had its own zeitgeist. Remember from lecture 1, it had its own culture and attitude that invited a scientific understanding. So experimental physiology was recognized as a valid research area. So you're going to hear about Weber and Fechner and Helmholtz, all German, because you could study this and you could be recognized as an expert in this area. We also know that the German spirit and the Swiss spirit, as we modern Switzerland, they put a strong emphasis on precision or exactness. There was a very important need for accuracy. So this is going to bring researchers to areas of zoology, biology, and physiology. Our primary interest in this lecture today is biology and physiology. But when we talk about Charles Darwin, we start to become a little more interested in zoology as well. Now, another cool thing about the German spirit was that they operated based on inductive reasoning. They started with a curious mind, collected the data, and then only after they had sufficient evidence did they formulate a theory about whatever it was. Now, we know that there's inductive and deductive reasoning. There's a lot of research that operates starting with a general theory, and we collect data afterwards. But one of the benefits of starting with an inductive reasoning mindset is that you don't go in with a bias. You go wherever the data goes, sort of like a journalist. If you ask a journalist, they don't know where that story is going to go. They follow the arc of the story, and wherever it takes them, it takes them. So that in terms of the German spirit, inductive reasoning, they followed wherever the data took them. So they also had this radical acceptance of biology, and biology was considered appropriate. And in terms of science, they were well ahead of the National Science Foundation, right? In the United States, we have the National Science Foundation, which defines what science is. Their understanding of science in Germany included things like history and linguistics. They viewed them as forms of science, whereas in the United States, the National Science Foundation, we had to fight for recognition, even as psychologists who do experimental research, to be recognized as a legitimate science. And you're like, well, who cares if psychology is called a science or not? We do, because there's a tremendous amount of funding that we would be excluded from the STEM grants, that just because of being arbitrarily labeled not a science, we were not eligible for. And it's absurd, right? At this point in your psychology career, you're probably sick of hearing about this research study and that research study. Everything is built on the foundation of science. But in Germany, very broad. Things that we might consider softer sciences were considered science in Germany. Now, you also had this movement in academia, right? So the German universities had certain rules. So there was this academic freedom. So you could teach freely what your scholarship was. You didn't have to filter your scholarship. You also could teach the way you wanted to teach. Now, academic freedom, this concept has permeated American institutions as well, but it started in Germany, that a professor has the liberty in how they teach. So if you took the history of psychology with a different professor, maybe they would teach it differently. But we have academic freedom to teach it the way that resonates with us, and we believe will help you learn best. So you also see this push for professorships. So research and professorships, faculty positions, were considered elite. And in Germany, they wanted as many scholars as possible. So what does that do? That creates increased broad base of knowledge and science that other parts of the world were not doing. They also had, better lab equipment, and they got paid better. So if you think about professors, as a professor, I get paid far less than when I'm doing therapy, but I love teaching, right? There are places in the world where professors get paid even starting salary 6 figures. That doesn't happen in the United States. But in Germany, that mindset was very powerful. They wanted to make sure that their professors could do their research without having to worry about finances. That's going to invite you to spend more time in the lab and develop more scholarship. Now, if you're wondering other places in the world that currently adopted this mindset for teachers and professors, go to the Scandinavian countries. Their professors don't get paid better than in places like Norway and Sweden and whatnot, because their mindset is very similar to this German approach. And then another thing that we have is a mindset of publish or perish. There was a pressure, and you remember Mueller was publishing an article every 7 weeks, but there was a pressure in Germany, publish or perish. What does that mean? If you stop your scholarship, then you're no longer useful in the discipline. You're going to start to teach in antiquated ways, and you're not going to be in tune with the latest and the greatest. But in order to get tenure in the United States, you have to have a certain amount of scholarship, certain amount of research, certain amount of publication, certain amount of commitment to the discipline, and so forth. And this comes from Germany. So this mindset. So what's the net effect of all of these expectations and the zeitgeist? Is that in Germany, they had the best professors. They had some of the earliest important discoveries in psychology, and ultimately they become the first location that invites the growth and development of our discipline. So we'll get to Wundt, but this spirit was part of what's going on. Now, how am I doing on time? Okay, I'll be able to probably get through Hermann von Helmholtz, but we'll probably have to stop in the middle of this discussion. So Helmholtz, as you'll probably hear him, German physician, born in Potsdam, Germany, was very sickly as a child, ultimately gets a medical degree from Berlin Medical Institute. And they had a program that if you were to be a surgeon in the army, you didn't have to pay for tuition. So he capitalized on that and spent 7 years as an army surgeon while he continued to do his research and study of mathematics and physics. And while being a surgeon, he's publishing along the way. And he was able to mathematically formulate the law of conservation that we talk about in physics. So to be able to go from the theoretical understanding of the law of conservation to the mathematical computation of the law of conservation is pretty incredible. So ultimately, he gets on faculty at various institutions. So Heidelberg, Bonn, Königsberg, and then eventually serves in the physics department in Berlin. Now think about that, right? He's in a physics department, even though he's a physiologist, he's a medical doctor, right? So the emphasis on the mechanistic spirit of connecting physics to our brain and body was very powerful. What you tend to see is that he was curious. I referred to him earlier as a Renaissance man. So he had so many interests, so many areas, and he published in many, many different areas. And ultimately, that resulted in him getting a lot of attention and put him on the fast track to what we say is fame. So what did he contribute to? If you were to think about his contributions to the field, the ophthalmoscope, right? So if you think about the light to look at the back of the eye or to check the retina that your primary care physician uses, he invented that. When we think about the Handbook of Physiological Optics, he wrote a 3-volume set on vision, which eventually gets translated. He had a theory about hearing and understood our functioning of hearing and developed a theory based on it. And then he wasn't done yet. He also wrote on hay fever and glaciers and geometry and the Arabian Persian music scale and the radio. So when I say he had a lot of interests, it wasn't just psychology. It wasn't just physiology. He was an absolute Renaissance man, a person who knew a lot of many disciplines. So here's an electromagnetic motor. So if you were to think about how we generate electricity, so he created this electromagnetic motor to power his lab. So you're seeing his inventor skills. So let's get to his studies on physiology. So if we were to think about neuronal conduction or the rate of neuronal conduction, he studied a neuron. He isolated a neuron in a frog's leg muscle, and then he stimulated it, and he was able to calculate speed of conductance, which he concluded was 90 feet per second. And if we were to talk about electrical impulses, we want to study the speed of a neuron, because it has a lot of important applications, things like multiple sclerosis. One of the understandings is that when you have an unmyelinated neuron, impulses move slower. So the impulse may not get to its destination. So understanding the speed of conductance is important. So he also suggested that thought and movement were not simultaneous, which was true. There is a reaction time, right? So remember what we talked about reaction time. That's true. And when we talk about studying reaction time, he understood individual differences. So you're not going to get identical outcomes when you study one person to the next. So when we talk about vision, he also understood the eye muscles and formulated a theory of color vision known as the Young-Helmholtz vision or the trichromatic theory. So if you were to look at the electromagnetic color spectrum, he said there are 3 primary colors in the eye. You have red waves, you have green waves, and you have blue waves. And it's the combination of these 3 color waves that result in producing any color in a color spectrum. So the trichromatic theory of vision is one of the prominent theories of how we understand cones or color vision. So just to give you a sense, you might have a receptor, a cone that's more sensitive to the red end of the color spectrum. You might have a receptor that's more sensitive to green or blue. And that's his trichromatic theory. And the combination of these is responsible for creating our perception of color. So he also had a theory of hearing. So our perception of tones, our ability to understand harmony and discord, meaning sounds that should appropriately go together and sounds that shouldn't go together. And he ultimately applied this to what's called the place theory of audition. And basically he suggested that in the cochlea, the sound, depending on what it was, is going to strike different parts of the cochlea. And when it's on a certain part, it's going to be a softer sound. On another part, it's going to be a louder sound, and it affects frequency. So the shorter the fiber, the higher the frequency. The longer the fiber, the lower the frequency. And it's sort of like strings on a piano that vibrate and create sound. And guess what? If we look at the cochlea, sure enough, it is mapped out in terms of cycles per second or hertz that we hear things, and it is mapped from low to high frequency. This is a good place to stop. I'm going to stop here because actually maybe I'll cover this. The basilar membrane of the cochlea, we have high frequencies. The smaller it is, the higher the frequency. Lower frequencies are longer strings along the basilar membrane. So I'm going to stop there, and maybe I'll do a running start with Helmholtz again. But I think it's important to note that Helmholtz, while not a psychologist, really shaped a lot of our sensation and perception, a lot of our neuroscience, a lot of our cognition research, and he's pretty well cited in our discipline. So let me stop my share, and then I'm going to stop the recording, and I'll take attendance one more time.